welcome everybody to the new chapter of the DY Patil Postgraduate Lecture Series. We have a very, very interesting talk today by none other than Dr. Sydney, who I'm sure all of you are familiar with from his days in Nimans. He has since migrated to Australia and now he is at the Queensland Mental Health Services. But I will leave the introduction part to Dr. Murthy. But welcome to the platform, Dr. Sydney and Dr. Murthy, both of you. And I thank you for making the time and being with us today, despite the time differences for Sydney and the busy schedule that both of you, I'm sure, have. But we are grateful to have you back on our platform. And I'm sure a lot of people are looking forward to this discourse today. I have the privilege of introducing Dr. Murthy to you. Dr. Moirangthem is an associate professor of psychiatry at the Krishna Institute of Medical Sciences at Karad. She has done her MBBS from JFM's PM Medical College, and she's done her her MD in psychiatry from GS Medical College, that's KM Hospital, Mumbai. She has been providing holistic and ethically rooted medical services for several years. As a teacher, she loves to engage with students in a manner in which probably she can help them understand the basics of psychiatry, but beyond that, generate an interest in the subject as a possible career option as well. For undergraduates, of course, and for postgraduates, she is a teacher who probably would inspire them with not just her repertoire of basic skills, but also her experience of many years that she brings to the table. Her interest lies in areas of consultation, liaison, neurosciences, and education. And she says that she enjoys learning as much as teaching. So, besides these, she has a host of other hobbies and activities which only go to tell us that she is a very, very multifaceted person beyond the realms of psychiatry. So welcome to the fold, Dr. Murthy, and I hand over the proceedings to you, and I'll come back at the end of the program to give my vote of thanks. Over to you, Dr. Murthy.
Thank you, Dr. Asmin. I'm very grateful for this opportunity. Talking about the neurobiology of suicide, I think suicide is one of the most studied behaviors, or one of the most mysterious and frustrating components of psychiatric practice. And even though there has been a lot of talk about how to prevent it, we know very well that not every suicidal act can be actually prevented. And within the context of the biopsychosocial model, the psychosocial elements are quite easy for us to absorb, but it's the biological that really gets to us. It looks like a lot of alphabet and numerical soup with a lot of different kinases and a lot of different areas of the brain that are getting involved. So I think it is a difficult topic. PGs have to be aware of this, and I'm really happy that Dr. Sydney is joining us today to talk about the neurobiology of suicide. I'll briefly introduce him. He's a specialist medical officer, psychiatrist at the Central Queensland Hospital and Health Services in Queensland. He is a former additional professor at NIMHANS, Bangalore. His areas of interest include bipolar disorder, epigenetics, emergency psychiatry, forensic psychiatry, and telemedicine. In terms of training and education, he has completed his postdoctoral fellowship from University of Gainesville in Florida. He did his MD from the prestigious PGI Chandigarh. He completed his MBBS from Regional Institute of Medical Sciences, Impal. He's affiliated with the Indian Psychiatric Society and is also was also a member of the Ethics Committee of this Advanced Center for Ayurveda in Mental Health and Neuroscience at NIMHANS. He has close to 770 public citations at present. His H index is 14 and I-10 index is 19. I welcome Dr. Sydney and I hand over the lecture to you.
Thank you very much, Dr. Murthy. Let me just start with sharing the screen. Am I audible?
Yes, you're visible and audible.
Okay. Are the slides visible?
Yes.
Okay. All right. I really appreciate Rasmim for this one giving me an opportunity to present this topic. In fact, we had decided a long time to do it, but last year I was moving from India to Australia, so we could not really do it. And he has the patience to give me a slot. So thank you, Rasmim. And I'm really glad to be doing this presentation. Uh, Before I start this topic, I will make a disclaimer. I did a similar presentation, I think around 2018, and was going to see the changes about what has really changed in the past 4 years. Lost. I'm not really sure how much has changed because the more you read, the more murkier you become. Something which was proposed becomes a probable marker, and then, like in every studies in psychiatry or psychiatric genetics, and the conclusion, or rather a definitive outcome, is very, very difficult to predict for a behavior which is a public health problem. So what I am going to do is I will just try to understand what are the models which were operating in suicide before, and what are the things which happens in some of the disorders we come across every day in our practice, which are associated with a suicidal behavior. Then we'll try to discuss some of the current understanding and try to go from there. Uh, the neurobiology of suicide will be an oversimplification if we try to discuss everything in this forum. It's too very vast and too very extensive. I have left out the proteomics and the omics part of it because they will be a discussion by itself. The epigenetics and the genetics part are mentioned as references because, again, they are discussion by itself. I've left out the imaging. So today what we are going to discuss is more like a condensed, maybe like an abstract for before you read a bigger paper. I might have missed something. Some of the data can be a little bit dated. If there are some error, please do correct me. And I would like this to be more like a discussion and an interaction rather than me just presenting and the rest are not contributing. So anyhow, let me start. Uh, there is a contest in whether the death by suicide is really increasing or really decreasing. In 2018-19, or even before that, the WHO said 800,000 deaths every year. Now they are saying 700,000. So it seems to suggest there is some decrease. But again, when we go for methodology and the way the data were collected, this might be a contested space. However, when it comes to India, there seems to be an unfortunate 2.5 times increased death of suicide in men. This is the data from the NBRC. And the red stands at 14.4 per 100,000 population. Suicide, yes, we deal with it. This is what we do, and understandably, the etiology is multifactorial and complex. As far as our understanding goes today, there is no one single mechanism or methodology or understanding of how a cell works that can explain a suicidal behavior completely. So we use genetics, epigenetics, neurochemical, neuroimaging studies just to understand the suicidal behavior. From what was a stigmatizing or rather a religious or a social event with no possible known reason, at least medically, we are trying to get some lead, or we are getting some lead. At least the way forward is becoming a little bit more clear than what was there maybe 10 years or 20 years back. As much as we want, when it comes to behavioral disorders, where psychiatry is the one, we are yet to find a biomarker or a set of biomarkers which can safely say that, okay, a person with this thing in the blood, in the brain, in the imaging or in the genes will be suffering from this. We are still not there. But nevertheless, the quest for something which can be a probable biomarker, at least for suicide, is, I think, a step forward going forward. So what have we been taught in the past that you need to have some suicidal, some factors which ultimately leads to a suicidal behavior. In India, we don't need to look at the factors. Down south, February, March, there is huge amount of student suicide. Up in the west, farmer suicide, again in down south. Then in the east, more like drug addiction, again complex psychosocial. And in the whole scheme of things, probably the component of a mental illness contributing to suicide in the whole scheme of things may be probably less, but nevertheless it becomes a significant genetic component or significant component. So what we have understand is maybe there is some genetic or some childhood traumatic events, some psychiatric disorders, which can be in the event of a stressful situation and the so-called diastasis can ultimately result in a suicidal behavior. This is basically what we have been taught for so long as biopsychosocial model tries to explain in that form, but it just didn't fit. So again, the same thing. It is the diastasis. What exactly is happening? So why do some individuals who face the same situation as the victim does not commit a suicide while the victim does? Simple example of social issues may be uh, the suicidal behavior in school going children. I mean, when the 10th exam or the plus two is the most stressful. 100 students in a class, maybe one has a suicidal idea or less than one will attempt, but what not the remaining who face to the same situation of question or difficulty. So, so this is something we try to understand. Yes, the same thing if we have to talk early life adversity, epigenetic mechanism, blah, blah, blah. I mean, this is what has been understood so far to some extent. The whole of psychiatry is based on neurotransmitters, monoamines in the beginning. We have moved away from there, but still not away. But so somewhere down the line, the hypothalamo-pituitary adrenocortical axis still seems to be. So this is basically what is or what are the risk factors. I will start with some of the common disorders where we see suicidal behavior in our patient, and I'll try to explain something about them. Then we'll go into the individual pathway or the individual etiologies or potential etiologies as we understand. So let me start with bipolar. We all know the risk for suicide is 60 times than the general population. 25 to as high as 50% will happen at one suicide attempt their entire lifetime, and an unfortunate 10 to 20 will have a complete suicide. Earlier onset of the disease is associated with an increased risk. Longer duration of illness correlates with a higher lethality of suicide, particularly in men. The usage of antidepressant was very different when we start learning psychiatry some 20 years back, but today, as we understand, is antidepressants are not to be prescribed unless it is really warranted, because it increases the switch and it increases the risk of suicidality. Then what happens to this individual of bipolar disorder when it comes in terms of imaging? Is there something concrete or is there something very distinct by which we can say that, okay, an individual with bipolar disorder with this imaging changes in the brain is likely to commit suicide? No, it is not like that. These are all association studies. These are done in population, but what we are sharing today is a common finding, right? So when we do an imaging in patients with bipolar, there might be periventricular white matter and deep matter hyperintensities. Many studies have reported a lower gray matter volumes, particularly in the region of the brain, which are generally governed with emotion, cognition, then judgment. And when it comes to bipolar, particularly the ventrolateral prefrontal cortex and the dorsolateral prefrontal cortex, the right orbitofrontal cortex, the right hippocampus, and the bilateral cerebellum. These are areas where in follow-up studies, in cohort studies, there are reported to have lower gray matter volume. And what if something happened on the right, then something should be happening on the left. So lower left orbitofrontal cortical thickness and white matter fractional anisotropy. And lower volume in the left ventrolateral prefrontal cortices. The right dorsolateral prefrontal cortex. These are areas which are involved in executive function and voluntary emotional regulation, and the bilateral hippocampus. These things are most important because when we say suicide and there's a trigger or something of a social event, the trigger can be a memory. That is what hippocampus do. Damaged hippocampus or a not so functional hippocampus can gives rise to an event which can culminate in a suicidal behavior. One thing I'd like to clarify here is when we say imaging studies, these are done in populations, but reproducing the same thing in every other study becomes a challenge and they have a lot of limitations. Is there some genes which can be implicated, if at all if we can use the word implicated, in bipolar? The initial studies were on gene polymorphism. What we need to understand is psychiatric genetics was in infancy more than 30 years back. We are just going forward, and the understanding about genetics in bipolar or mental illness is limited by the techniques that was prevalent in basic genetics. So polymorphism was something of the early 90s or early 2000s, sorry, late 90s, early 2000s. So in gene polymorphism, there is a tryptophan hydroxylase and tryptophan hydroxylase 1 and 2, which are found to be related to suicide attempts and in terms of high lethality and completed suicides. These are, correlation, not causality. And polymorphism and low expression of the serotonin receptor RNA in the prefrontal cortex again has been associated with suicidal behavior. The techniques of doing a gene assay has progressed quite a bit. Initially, it was microRNA focusing only on polymorphisms. Now it is the DNAs of RNAs and microRNAs. So basically, this slide just gives a reflection about the change in the technique over the period of the decades that we try to understand suicide. And BDNF, which is the poster child of psychiatry, the expression is less in individuals who commit suicide independently. Independently means independent of any disorder, whether it is bipolar, schizophrenia, major depressive disorder, or otherwise. So what happens in MDD? Yes, the risk of suicide is 20 times higher. Prevalence of suicidal ideation is 37 times, and prevalence of suicidal plans is 15%. Slightly better than what it is in bipolar. So is there something of an inflammatory marker which can correlate suicidal behavior in depressive disorder? So there are some which suggest they might. There is lower BDNF, increased interleukin-1, interleukin-6, and TNF. And basically, they are all related to a dysfunction in the HPA axis, leading to an overproduction of cortisol-releasing hormone and elevated glucocorticoid production associated with structural and functional brain changes. We'll discuss the details later when we go into the inflammatory markers later on. Anxiety disorder is a contested space. Some people with specific anxiety disorder can have suicidal ideas or suicidality at least, but most of the studies show that the role of anxiety in suicidality remains unclear. Only one anxiety disorder which are clearly correlated with suicidal ideation or an attempt is OCD. And it has been shown that one patient out of 10 has a lifetime suicide attempt and nearly half of them have suicidal ideation. So let's come to the major psychotic illness or severe mental illness. Contrary to popular belief, a schizoaffective disorder has the highest risk of suicide attempt and completed suicide, followed by schizophrenia, first episode psychosis, and the list among the orders seems to be delusional disorder. Yes, we might contest by saying that do schizoaffective disorder really exists, or is it an artificial construct? So that is a discussion for another day. So, but this is what at least the study of some of the evidence shows. So in schizophrenia, there's a lifetime prevalence of suicidal ideation, which can range from 20% to 35%. Lifetime prevalence of suicidal attempts and self-harm is as high as 26%, and nearly a quarter of the patient has a complete suicide, which is very, very unfortunate. First episode psychosis is an area which is being explored so that, with the belief that a disabling long-term illness can be truncated. We're not still successful. We are trying to. And 40% of the patient can experience persistent suicidal ideation. When it comes to the psychotic illnesses and the risk for suicide, numerous studies have been done right from the beginning, as early as 1980s and 90s, from trying to understand the HPA and the glucocorticoid neurotoxicity, then trying to measure the CSF level of serotonin metabolites and blunted response. So far, this pathway are still valid, but it's just that the relevance is not just in this pathway. So what does the evidence suggest? So there is a decreased expression of the level of two glutamate-related genes. That is the glutamate ammonia ligase and glial affinity glutamate transporter member 3. A very, very long name with a abbreviation which is very different from what it sounds. And these are found in individuals with schizophrenia who has committed a suicide. The study which I quoted that does not differentiate whether it was single attempt, multiple attempt, or a completed suicide. But since this is a decreased level which relates to the epigenetics, this is again a contested space. There are some evidence that polymorphism in the adrenoceptor alpha 2b is associated with suicidality among patients diagnosed with schizophrenia. All the while we are talking about association. In statistics, it's not causation. It is just that the presence of these events may correlate directly with the severity of certain thing, or they are related, not necessarily this cause. So we should be very careful and not to conclude very prematurely. Borderline personality disorder is something, at least if I have to share my experience while I was there in India and now, yes, we have seen borderline in our outpatient as well as inpatient in our Indian practice, but they are not very prominent. We see more psychosis, more mood disorder, but trust me, here how Australia is flooded with BPD. They consume 50% of the ED services, and it's very, very taxing. And truly, when we try to understand personality disorder as a construct, again, this is a contested space. Whether you see at genetics, again, difficult. But what is the latest, at least in when we try to understand this cohort of individuals who presented with a suicidal behavior? So there's a 52-fold increased suicide rate in borderline compared to the general population, almost like the bipolar. 75% will attempt suicide, and 6% may die with suicide. Again, there is an association with the polymorphism of the serotonin synaptic system, particularly in men, and the serotonin transporter region in males. Basically, it just boils down to serotonin, and as much as we try to go with serotonin as this whole thing for mood or impulsivity, we are not just aware. Because somewhere down the line in the brain, in their mechanism of how it work or how they work, serotonin still plays a central role. But again, the central role is not sufficient to point that this is the thing which will happen and which will predict an eventual suicide. What about substance use? Substance use disorder and suicide are, we can say, two-way bidirectional traffic. There will be a group which says that substance use increases the risk of suicide, which is also true, and hence substance use does it. The other group will say just the opposite, because there is a vulnerability, a disorder. So alcohol or substance will be something which people use to control it, or at least to some extent. And it is bidirectional. Alcohol, as we understand, is one of the most used substance after tobacco. So hence it is associated with a 94% increase in death by suicide. When we talk about opium and cocaine, the evidence is very faint, but some polymorphism related to the BDNF gene has been found to be associated with suicide attempts in these users of opium and cocaine. And now the latest understanding of substance use suicide is when it comes to both the imaging as well as for the cells, there is an alteration in the expression of genes involved in glial differentiation and glutaminergic transmission in the dorsolateral prefrontal cortex, which are observed in patients with a suicidal behavior with substance abuse, irrespective of the substance. So it all boils down to the area that is the dorsolateral prefrontal cortical area in most of the suicidal behavior that we have discussed till now. So basically, just a summary about what are the molecular changes found in the brain of suicide victims with a psychiatric disorder. So what happens in schizophrenia, what happens in MDD, it's just a summary. Something which is very important when we talk about prevention, or rather primary prevention in mental health, is child sexual abuse or child abuse. And there is a strong correlation between child abuse, early adversities, and the risk for suicide. The National Epidemiological Survey on Alcohol and Related Conditions, which was done for a different reason, of course, to tap into early life adversities when they try to analyze whether adversities as a vulnerability is a risk for substance abuse, then a lot of respondents reported childhood physical and sexual abuse. And when it comes to child abuse, most studies have found that children, adolescent, and adult exposed to abuse or neglect during the childhood have 2 to 4-fold increase in the risk of suicidality, which includes ideation, plans, and attempt. And among all the childhood adverse events, child sexual abuse correlates most strongly with suicidal behavior, which is quite understandable. So why does early life adversities leads to a later increase in suicidality? Some of the explanation which has been put forward in our understanding is that early life abuse leads to persistent hyperactivity of the CRM. Then what results is that it sensitized or hypersensitized the pituitary, adrenal, and autonomic response, leading to a persistent or a heightened stress response. This is the basic early understanding. Then in imaging, childhood maltreatment, they are associated with lower gray matter volume, decreased cortical thickness, and hypoactivation in the ventral, medial, and the dorsal prefrontal cortex, the exact same area we have talked before, including the orbitofrontal and the intercingulate cortices, the hippocampus, the insula, and the striatum. And some of the latest studies, particularly when it comes to the white matter, is there is a disrupted white matter structural integrity, which then leads to cognitive impairment and the disruption of emotional regulation, thereby increasing the risk for suicidality. Here we are not talking about a developed brain. We are talking about a developing brain, a child where the brain is still growing. And when there are so many disruptions, then we cannot expect the trajectory to miraculously disappear. So this explains why there is cognitive impairment and why there is a disruption of emotional regulation. So this also partly explains some of the emotional dysregulation we see in individuals with borderline personality disorder with a history of childhood adversities. Okay, what does the genes about childhood adversities says about? Early studies, there is polymorphism of genes. Again, what are the genes? It is the corticotropin releasing hormone receptor 1 and 2. Then there is a protein with a very long fancy name, that is F506 binding protein, which is a chaperone for the earlier one, in which there is polymorphism. That means they are not a normal variant. They are slightly the different one. And the corticotropin release binding protein, the CRHBP. And the polymorphism in this correlates with childhood maltreatment, suicidality. It is just a correlation. And if we have to just visually try to understand what happens in childhood adversity, is there is an early life adversity which increases anxiety due to alteration in the SP axis. Then there is epigenetic alteration, which is mainly the pathways of the RNA, and which can potentially give rise to a depressive psychopathology and suicidal behavior. So this is basically the summary about what could happen or what will be the pathway of or the outcome of childhood adversity and suicidality. All right, so let's go to the pathways of or the pathways which we understand or try to understand our mental illness so far, the hypothalamo pituitary adrenal axons. And yes, early life stress are associated with aberrant HP axis in adulthood. Then again, childhood trauma increases suicide ideation, which is we have just seen. Many of the early studies which was done in postmortem samples, they use the frontal cortex, the cerebellum, and some studies also use the amygdala samples because we believe the amygdala is the the seat of emotion, which is not exactly true. I mean, it's just one of the regulators. So when amygdala samples were used to analyze change in the fancy protein KB5P and nuclear receptor family 3, 1, I mean, it's a long fancy name. They all deal with glucocorticoid receptors. So they are found that there is a significant reduction in the target gene and protein expression in the brain region, particularly in the amygdala. However, when all the meta-analysis, I mean, the past couple of years, I think the significant one good one came out in 2023, another 2021, 22, and a couple of them before. I mean, a lot of studies were done, but when it comes to the final conclusion, excluding mental illness, independence, suicide, alcohol, no matter how much there has been noise about HPA, there is no hard evidence for HPA axis dysregulation, suicide, and early life adversities. So this is the difficult part about the psychiatric research, because a lot of factors can affects the outcome or even the reproducibility. I mean, the population genetics variability is one, the comorbidities are another, substance abuse is another, and even the way the techniques have evolved and interpret they all make a definite conclusion about the HPA being completely dysregulated becomes a little difficult. But this is how science and medicine progress. So psychiatry is no different. So, but we go ahead with the data we have. Yes, neuroinflammation. I mean, the poster child or the print child. There is higher concentration of chemokine, some fancy name CXC motif, in serum of patients who has a risk for suicide. Just give me a minute. Apologies. And in this patient, there is higher suicidal risk with early adulthood adversity, but again, not with early life stresses. So this is, I mean, we can call it here a very honest research in the sense that, I mean, when you do an initial analysis about, let's say a post hoc analysis, okay, the earlier sample shows this, but post hoc doesn't show this. So yes, neuroinflammation, some of the ligands in the serum which are high or less is associated with adult, but it's not associated with early life stress. How do you differentiate that? Difficult question to answer. So honestly speaking, I don't know, because none of the studies which I have referred have tried to explain why or what was what are the indicator, or how do they screen out, or what are the criteria by which the adult and the early life were excluded. But this is how it is. However, when it comes to neuroinflammation, the cytokines are we can say the in thing in the sense that neuroinflammation and mental illness, it is the understanding has improved, and it is not just related to suicidal behavior. It is related to mood disorder. It is related to major psychosis like schizophrenia. So when it comes to suicide, uh, there are some cytokines which are consistently found to be elevated, depressed, and hence associated with suicide. So they are interleukin interferons and tumor necrosis factor. But if we look at these things, the interleukins, the interferons, and the TNF, they are inflammatory markers in acute inflammation, the presence or the absence of which may result in an acute inflammation resolving and healing of the tissue to going to a chronic inflammation which becomes detrimental. So it's no surprise they do the exact same thing when it comes to the brain. So they are developed from the thymus, and there are two lineages, TH1 and TH2, and some are pro-inflammatory. That is the 1, the 6, the TNF alpha, and the anti-inflammatory will be interleukin 4, 10, and 13. The most consistent cytokine which is found to be associated in suicidal patients is an elevated level of interleukin 6, either in CSF or in blood. After that, it is the TNF alpha, which is found in higher associated to suicidal behavior, suicidal ideation, or even a complete suicide. The interleukin 2 and the interleukin 2 soluble receptor are found in some studies to be lower in depressed suicide attempters than depressed non-suicide attempters and healthy control. So these are set of, you can, or rather we can say triple blind, if not exactly blind, that they have taken healthy control, depressed individuals who are again differentiated into suicide attempters and non-suicide attempters. So even in the same set of individuals who are depressed or possibly suffering from a mood disorder, the interleukin 2 levels were lower in the same population. So this goes on that, yes, maybe it's a little more specific to say that interleukin 2 level can it be a probable biomarker? Yes, I mean further studies are more samples. Interferon gamma is very non-specific. The latest to join the queue, or rather the family of cytokines, is the vascular endothelial growth factor. All the studies do conclude that there is a decreased level of vascular endothelial growth factor in areas of the brain of interest to the investigator. So these are important areas of interest of the brain to the investigator, because not everybody studies the whole brain and tries to study every region of the brain with the same marker or same cytokine. I mean, they just pick up one region and try to answer the question which they have asked. So what it means about the vascular endothelial growth factor is when we have a decreased level, the angiogenesis stops. When the angiogenesis stops, the neurogenesis halts. When the neurogenesis halts, the neuronal plasticity stops. When all these three happens, the repair simply stops. When the repair stops, the suicidality increase. So these are actually very, very hard evidence that cytokine, or the presence or the absence of which are very, very significant in an individual with suicidal attempt. But again, when we look at suicide as a whole, inflammation or neuroinflammation has a very, very strong evidence, but it's not able to explain the entire spectrum of suicide. But nevertheless, these are very, very strong areas. The neuropeptide and the suicide was, we can say, the PS3 discussion maybe 10 years back, when we talk about so many things, vasopressin in the early 1890s. Initial studies seems to suggest that yes, there is a role. but the subsequent meta-analysis systemic review proves that there is hardly any correlation. Sad, but this is how we progress. Now oxytocin is coming into some relevance, and the conclusion that we can draw about oxytocin is that the concentration reduces in suicide attempters. And yes, when we talk about neuropeptides, we start with vasopressin, we go to oxytocin, then we also look further. The latest, or rather one of the newer ones, is the neuropeptide Y, which is also referred as a resilience-related peptide. If it is resilience-related, that means we expect it to be on a higher side or on a higher level to be not suicidal. And understandably, when there is a suicidal idea attempt or a completed suicide, then there is a significant decrease in neuropeptide gene expression and increase in the expression of this receptor, which is quite understandable. The receptor expression will increase because you are not getting the neuropeptide, and you will try to get whatever neuropeptide is available. Again, in the region of interest is the hippocampus and the PFC. Psychiatry had a lot of growth with serotonin. We are still there. We're still going further. A lot of noise around it, but again, very, very inconclusive. But nevertheless, when it comes to suicide, it has been rather found or correlated that patients with schizophrenia and depression, in their dorsal raphe nucleus, where the primary neurons innervate the prefrontal cortex, there is a decrease in the total number of serotonin receptors. So what do the serotonin transporter do? It is responsible for sodium-dependent serotonin reuptake. And the 5-HT gene, if we just have to go for our academic understanding, is in the located in chromosome 17. As much as it looks fancy and loud all through the years, whether serotonin levels are high or low in depression, or whether antidepressants are still working or not working, there is no concrete evidence to point that serotonin solely is the system which is responsible or implicated in suicide. Then next is the endocannabinoid system. The endocannabinoid system is not just limited to suicidal behavior. These are potential targets in psychiatric disorders because it regulates emotional response. It is becoming extremely loud, or rather sometimes little biased, in the sense that there is a lot of industry pressure to promote opioids, cannabinoids, either natural or synthetic. So understandably, there will be more studies, or at least some studies, which will try to say that whether these things or the mechanisms are of any benefit or whether they can be targeted. So for our understanding, what we need to understand is that there is high frequency of SNP of the CBR1 receptor gene in patients with MDD and schizophrenia. And a CBR2 receptor in the CNS has a role for possible anxiolytic and antidepressive target. All the cannabinoids genes or endogenous one, they are G protein coupled receptors. And one of them is the GPR55, which are targets of the endocannabinoids. And in some of the animal studies, particularly in mice, the result is that stress-induced changes, some change in the gene expression. I mean, it's not going up. That means the proteins or the endogenous cannabinoids, they're not coming up. So again, which region of the brain in the mice? So again, the dorsal prefrontal cortex. When it comes to alcohol dependence and suicide, again, because substance abuse is, as we said in endocannabinoids, it's not just it's becoming quite significant in psychiatric disorders. So substance abuse, particularly alcohol. So there are a lot of studies, and a common finding is that there is an elevated level of anandamide and palmitoethanolamine in serum of suicide attempters of alcohol dependence. So far we were dealing with some of the cytokines, some of the endogenous system of neurotransmission. So let's try to understand the epigenetics of suicide. Why I'm not going too much into details about the genetics of suicide is, one, we have discussed a little bit, because, and secondly, the earlier studies in either GWAS or all those things have not found anything very, very significant. So people have tried to look, is there anything around the genome, not the gene, which can be responsible for some of the events in a suicidal attempt or suicidal behavior. So epigenetics tries to answer some of the things. So when it comes to epigenetics, the most studied modification is a DNA methylation, and DNA methylation is always repressive. It usually happens at the promoter region, because when the promoter is bound tightly, then there cannot be any translation. So it stops the gene expression. So this is what DNA methylation is briefly about. So when we look into the history of epigenetics and suicide, like in the beginning, like I've said in the beginning, that our understanding about mental illness or behavioral this modification or a set of behavior is directly proportionate or is directly correlates with the technique or the hypothesis that were proposed or ongoing at the time. So understandably, the epigenetics also start with a monoemergic system, like we discussed, serotonin being example, HPA being an example. But over a period of time, as understanding improves, now the focus has changed into other molecular pathway like the noradrenergic, glutaminergic, the GABAergic transmission, and newly the polyamine system. But when we look at the noradrenergic and GABAergic system, it is not just with suicide. These things are also gaining a lot of significance in schizophrenia as of today. So what happens to the GABAergic system? There is a hyperactivity of the DNA methyltransferase in the frontopolar region, and it leads to a hypermethylation of the GABA A subunits. Please understand, hypermethylation means the GABA subunit is not going to come out, right? And in the suicide brain, this leads to a downregulation of the GABA A subunit. Then it leads to a deficient expression, and the end result is the underserved hedonic and decision-making capabilities, which is impaired in the prefrontal cortex, ultimately resulting as an impaired cognitive process. So let's talk a little bit about polyamines. So they have come in the past 1 decade or so, and basically they are aliphatic molecules with 2, 3, or 4 amine groups. Some of them are aguantin, tryptophan, spermine. And what do they function? They function as neurotransmitter by binding to the NMDA, acetylcholine, and serotonin receptors. And what do they do then? They have some antidepressant, anxiolytic, anticonvulsant, anti-inflammatory, and neuroprotective properties. Every one of them is important, particularly when we say about antidepressant action, because there are a lot of papers which have come out that serotonin is not a molecule or serotonin is not changed in depression. Obviously, it's not going to change because there is a link molecule. What is actually working at a different level is polyamine is one of them. So we are not looking at where we should be looking. And people who doesn't like psychiatry make a lot of noise about it every 5 years or 6 years, and the cycle repeats. But the truth is different. It's just that our understanding about drug mechanism, the molecular mechanism, the genetic the genetic mechanisms are becoming better. The drug is the same, but our understanding is becoming better. So in the polyamine system, there is one particular gene which is known as SAT1, which is used in synthesizing spermidine, and it is a rate limiting enzyme. So why it is important? Because the promoter or region of this gene has a DNA methylation and downregulation in 3 Brodmann areas, B4A, 8, 19, and 11, in suicide completers. So these are more stronger evidence that there is actual some epigenetic changes in suicide completers. But again, when we talk about how consistent is the data to say that okay, this should be should be a biomarker and can it be done with everyone, then this becomes a very difficult question, because first of all, when we do a epigenetic or a genetic study, it is not always from a postmortem brain. We also use peripheral markers, which are proxy markers like blood, saliva, hair, skin. Yes, they give a very similar finding, but when we use peripheral markers, there are a lot of biases. So we are not in a position by which those biases or other difficulties can be removed and be concluded very firmly. But nevertheless, this is one way forward. Sorry. Okay, all right. Then again, let's go to the neurotropic system. The hippocampus has always been involved in the early studies along with the serotonin, because the I mean, these are areas of the brain which are believed to be associated with memory, and traumatic memory starts a loop of cognitive distortions which ultimately culminate in a suicidal behavior. And understandably, when serotonin starts, it will be correlated, as we understand before, that the dysfunctional serotonin system will be correlated with suicidal behavior. So again, when it comes to the region of area of interest of the brain, it's the orbitofrontal and the dorsolateral prefrontal cortex, which are found to be structurally abnormal in suicide victims, which is an indication of impaired neuronal plasticity. BDNF is the poster child of psychiatry. It is involved in neuronal growth, differentiation, and maintenance. As much as it is a non-specific marker, it seems to be involved everywhere. I mean, as much as it is non-specific, but it's involved everywhere. And in the adult brain, it plays a pivotal role in shaping the structural, morphological, and synaptic plasticity. And TRKB, or tropomyosin receptor kinase B, is a cognate receptor for BDNF. In patients with suicide, there is a diminished expression of both BDNF genes, BDNF and TRKB genes in prefrontal and hippocampal neurons, which is quite understandable, because when there is a decreased expression, then you will not find in either BDNF or TRKB. The Wernicke's area of the prefrontal cortex, that is the Brodmann's area 21 and 22, they are areas involved in the semantic thinking and decision making. And these are also mostly affected in suicidal behavior. And hypermethylation of exon 4 of the BDNF leads to this functional impairment and neurocognitive processing in a suicidal brain. For our knowledge, the BDNF has 9 exons, or rather 11, depending on how we study it. Exon 1 is one of the most famous, and exon 4 is now beginning to correlate with the suicidal behavior. But exon 4 is also believed to be, in a way, not related to our topic, but our preference for food between carbohydrate and fat. It is very difficult to accept, but trust me, I mean, the exon 4 is, at least in any animal model, correlates with a person's choice whether we want more fatty food or whether we want more carb-rich food. All right, yeah. When we say epigenetics and when we say DNA methylation, we are not talking about DNA methylation in the entire brain or in the entire neurons, or it's not like that. DNA methylation is just a process by which certain genes are suppressed, and the gene can be of multiple types. So when it comes to the hypothalamus or the HPA axis, there is a dysregulated glucocorticoid receptor. And when there is a dysregulation, it dampens the negative feedback regulation mediated by the glucocorticoid hormones, ultimately leading to an hyperactive HPA response under stress. And they are not linked with childhood adversities. Why it is happening? Because there is an altered DNA methylation profile in some of the studies in the glucocorticoid promoter in the hippocampus. Again, memory. So it is at this time, it is not difficult to understand that the DNA methylation that we talk so much as one of the important processes in epigenetics is the one which is working when we talk about the protein or the receptors in the brain or in the pathway that is of consideration in the study, right? So it's not very difficult. Initially, the RNAs of that we thought we knew, or rather, we thought it is important are our messenger, ribosomal, and transfer tree. Then our understanding of RNA is that it comes from a DNA, which is double-stranded, and one of the strands become an RNA. We forget to look at what happens to the other strand. So there is a new area which we understand as microRNA. They are non-coding but regulatory RNAs. And based on the type, they are either replace the translation and they sorry, they can inhibit the stability. So microRNAs are basically, if we look at the size, they are just 21 to 22 nucleotide big or long. And please do understand, a double-stranded DNA, one strands become RNA. The other strand, they also do a lot of work. So they are specialized role in neural development, maintenance of mature neural traits, synaptic and neural network plasticity, and memory function. And the microRNAs, they are expressed by number, and there are quite a lot of them in the neocortex and the cerebellum. And the microRNA is not just associated with suicide. In bipolar disorder, particularly mania, microRNAs, some of them like a 134, they are going or likely to become a very, very viable marker. But the difficult part is the harvesting the microRNA is not very easy. So when we talk about epigenetic mechanism, please do understand that the gene has a promoter, then you have the coding region, then you have the non-coding region, where in epigenetics, or rather the processes, the DNA methylation works, but there are also processes other than the DNA which are involved. You have histones, then you also have microRNAs. And we have talked about high-speed dysregulation, then glucocorticoid receptors and DNA methylation being suppressive. When DNA methylation is happening, by the by, the microRNAs are also working. It is not a separate process. They are not separate. They are all linked. So when we talk in terms of microRNA, the the downregulation, or rather the expression of glucocorticoid receptor, when we talk of the SPXs, which is downregulated in depression, is also done by microRNAs. And here it is the 124A. And again, when we talk about microRNA, the region of the brain also depends on the choice of the investigator and what they are looking at. So many of the studies, they were done in the prefrontal cortex, and a consistent finding is that there is a microRNA downregulation in the prefrontal cortex, and this can be a molecular signature for depressed suicidal patient. So let me just conclude here. So what's the way forward? We need better understanding of suicidal behavior. Better understanding of suicidal behavior in terms of the neurotransmitters, the amines that we talk about, the genetic and the epigenetic process. Our understanding, we are just starting. We are not there yet. And once we do that, then we probably can prevent or think of preventing suicidal attempts, which is way, way far too fast. But as we go forward, then we can also work towards the development of a peripheral biomarker. And hopefully, in the future, hope for a personalized treatment which our patient really deserves. So the conclusion is suicide as we talked in the past half an hour, 40 minutes, is multifaceted, multifactorial behavior. Stress adaptation or impaired stress is believed to be a mechanism which is involved. Our understanding of the neurobiological mechanisms are maybe marginally better, and biomarkers can be a way forward. So this is one question which I would like to ask to our audience. In India or in any country where there is high risk of suicide in the young and the adolescents, what could be the probable reason for increased incidence of suicide in the teens and young adult with the onset of spring? Thank you. And these are some of the references I have used.
I'd like to answer your question. I think it coincides with the time that major exams happen.
Yeah,
More psychosocial.
that is the psychosocial. So what can more be the biological? What can be the biology? Or talk to
Yeah,
them
the
about the
biological.
biology. Yeah.
Does anybody want to answer among the audience? So we have some questions also. So let's see if somebody's answering what you have asked. Okay, so until somebody answers, maybe we'll take the questions. Yeah. So Dr. Debalina wants to know what is penfluramine test in schizophrenia. I don't know if it directly is relevant here, but please let us know.
This is basically one of the tests which we have talked about the response of the stress hormones, but it is more not very significant at this stage. I mean, this is one of the many processes by which we have evolved about our understanding.
No. Any more questions? Yeah, I have one observation that I'd like. I was wondering if so early life adversities resulting in epigenetic changes appears to be a very important element in the biology of suicide, or neurobiology of suicide. I was wondering if exposing young children or young children who are exposed to early adverse events, but if they also experience love, affection, and enrichment, so there may be an abuser or one particular element in their society, in their social circle that is an abuser. But exposure to love and enrichment from the other elements, does that reverse these epigenetic changes? Is there scope for that?
Okay, yeah, this is a very difficult, what you call, there is no one single answer for that, but I will try to explain how we come to this conclusion. The initial studies when it comes to imaging about child sexual abuse and imaging particularly of the brain, the hippocampus, the dental is the area which is very much affected. I mean, there is always a decrease in the hippocampal volume in the entorhinal cortex. So this is one consistent finding. Yes, that is just anatomical scan. Then the next question they ask is why it is happening. What is responsible for those degeneration? Where do we say there is a threshold risk between what is a single exposure abuse and series of abuse? All right. So that is when they try to understand what is there any genes around it, but it didn't explain. Then the explanation comes to epigenetics about the downregulation of the HPA axis or any of the glucocorticoid receptors or any of the genes. So, I mean, there is no straight answer about how many exposures are required, but nevertheless, when there is significant impairment in neuronal plasticity or the BDNF is so very low, at least what we try to understand is that the brain loses the power to heal. And this power to heal is also based on the vulnerability. The vulnerability is defined by are you carrying a polymorphism when we talk about genetic understanding that makes you vulnerable. All right. So, and when we talk about vulnerability and polymorphism, then it's about how many polymorphisms or how many genetic imperfections are you looking at, right? And let's say there are here in Australia they talk about intergenerational trauma. Initially, it looks like a little bit political, but as we try to understand, let me give an example. We have inheritance from our parents or from our grandparents about genes, but the Psychiatric genetics does not follow the Mendelian genetics. That means one gene, one model, or one disease hypothesis. It is multiple candidate genes, and the candidate genes, when they are expressed in the same generation, not every one of them express. Some of them, the expression becomes late, and we have also our repair mechanism. So the number of genes which are expressed, mind you, we have, if I'm carrying 10 genetic this one mutations in me, the same mutation is going to be in my bones, my liver, my red cells, my muscle cells, including the brain cells. So but are they expressed in the entire region of the brain, or they are differentially expressed in some regions of the brain? So that is where the real difference comes to, why some individual have some symptoms while many doesn't. So all the studies so far have tried to show that yes, more adverse events are correlated to a persistent suicidal behavior, which is again correlated to some epigenetic error. Understandably, if we remove the source or the event which is triggering the process, like affection, proper care, nutrition, yes, the repair mechanism should work. Right? Like in the final presentation that I have said, the RNA. The RNA that we understand initially were just three. We forgot about the second strand of the DNA, right? So all those things, they come into place and they have a repair mechanism, an inbuilt repair. So yes, changing the environment do play a role. Yes, to a great extent, it is again psychosocial, biosocial. I mean, yes, it is. But here we are talking about the milieu will be a cellular milieu, again governed by the environment around. So as much as we have tried to move forward, the role for the environment as a source of stress or a source of an imbalance, at least with the evidence that we have today, cannot be contested or thrown away, not just yet.
Okay. All right. So that means we can reverse some of those epigenetic changes with love and affection. To conclude, we
Definitely.
can.
To
Yes.
conclude, and
Yes.
yes, even impulsivity, and should I say some of the behaviors, like the suicidal behavior that we see in people, irrespective of the diagnosis, or even otherwise, let's say pharma, for example, right? Giving a lithium or an SSRI will probably decrease the suicidality. Lithium does, because lithium has the strongest evidence for bringing up the level of BDNF. At least that is what evidence say. Yes, there are a lot of confounders to it, but at least this is what evidence is now.
Okay. Here we have a question in the chat box. What's the difference in neurobiology of impulsive completed suicides, planned completed suicides, impulsive suicide attempts, and deliberate self-harm? That's the first question. There are more ahead. What's the
Neurobiologically,
difference in the
Naresh
biology?
bhai,
Yeah.
we have no answer for this. Absolutely no answer, because the neurons does not understand what is impulsive or a deliberate self-harm, because deliberate self-harm, or even what you call non-depressive suicidal attempt, these are terms we use for our convention, just to so that we can makes the management probably less easy. But at the neuronal level, at the genetic level, it does not make any difference. And truly speaking, honestly speaking, all the studies that are being done till now, they have not really tried to differentiate between which is an impulsive suicide or which is not an impulsive suicide, or what is an accidental suicide, or why do some people with depression really tries to kill themselves, where for people with borderline personality, the attempt could be very little, but the intent was not to die, even though the outcome can be the same. So these are some of the unanswered questions, and there's no hard studies to say that it's there. I mean, it's different.
Okay. Does anybody else have anything to ask or contribute? If not, then I think we can call it a day, and we can have Dr. Rashmin come in. I think Dr. Sydney, any concluding remarks you would like to give?
No, no.
Okay.
I
the
think, yes, Sydney, please go ahead.
Yeah, if I have to just answer the question that I have asked, this was a hypothesis, I think, proposed by a French or a Polish group, where they tried to understand why there is an increased incidence of suicide during spring.
Hmm.
What they have proposed is in spring, because with the winter just end, the wind speed builds up. The wind speed builds up, and there are a lot of environmental pollutants, pollen being one of them. It's carried quite a lot, and to vulnerable students or adolescent, it becomes a trigger. So they have even proposed that whether the exam can be changed from spring to either the winter or late autumn, just to see. Because I mean, they did a small study in a group, and it was quite evident that the pollen was a trigger, because these individuals were already susceptible. They were susceptibility. The pollen was a trigger. The not so good performance at the exam was the final straw. So it is more like a preventive thing which was proposed, but again, could not find anything very conclusive out of it. But it is our ideas which people try to ask some questions, try to see whether it answers some of the things which we are trying to understand.
It's an interesting thing, interesting concept.
Yeah.
Yes.
Very interesting.
Okay, so
Very
I
interesting indeed.
And there
think
is
So,
even a group which now says that because the evidence for neuroinflammation is so very high, I mean, the evidence is strong, then in addition to the usual treatment of our antidepressant, that we understand medicines as antidepressant or mood stabilizer, are we justified in using immunomodulators? If the interleukin level is very high in acute inflammation, the people use glucocorticoid left, right, and center. We use that in COVID. It gives miraculous result. So why not? I mean, these are some questions which people are starting to ask, whether we can also, approach our treatment protocol little different with some of the medicines we already have. Again, discussion for some other time.
very,
yes,
very
yes.
true. I think, Sydney, you've done an excellent job of giving us a very, very broad perspective of various aspects of neurobiology. And so far, at least during my training days, we were all stuck with all the old concepts of the psychosocial models and the psychoanalytic models and the psychodynamic models. But now we understand that some of those don't apply anymore. Some of them, even if they do, we have to see them through the lens of our newer advances in the biology of the brain and our understanding of the biology of the brain. A lot of new genetic tools, investigative tools have become available to us. So they've helped us and have a fresh pair of eyes, for the want of a better word. So it was really an eye-opening session in many ways. And I'm sure all the postgraduate students who are with us today, as well as some of the senior consultants, have noted down a lot of take-home points from this enlightening talk, and I'm sure it has sparked our interest in this new perspective. And honestly, Sydney, you've kind of dug your own grave by enticing us. So we will have to invite you over for many more sessions in the future because
Definitely.
you've really sparked our interest, and it's also really nice that you presented it so lucidly that more often than not, uh, the older lot like me tend to get lost in the myriad numbers and figures and terms, but you kept us all riveted right till the end, and I'm sure we'll want to have you back for more. And
It will be my honor. It will be my honor.
no, no, absolutely, absolutely. And of course, Dr. Murthy did a very nice job of walking us through the entire session. And a big thanks to all the people in the audience who made their time today to be with us and uh, learn from this excellent opportunity of Dr. Sydney enlightening us about the various aspects of the neurobiology of suicide. We continue to strive to bring you such lectures in the future, and we hope you shall be back with us. And I would be remiss if I don't thank Sun Pharma for their unflinching support. So thank you all once again, and I wish you a good evening ahead, and I hope to see you all back again next month with another nice, interesting talk. Thank you, Dr. Sydney, and thank you, Dr. Meghna. It was very kind of you to make the time, especially Sydney is very late in the night for you. So I wish you a good night. And
Thank you. It's just 11. It's all right.
it's
It's
11.
just 11. We are four and
You're
yeah. It
You're an
We are
Indian
four
at
and a half
heart, so
hours
you are
away.
spoiled. Okay. All right.
I
Anyway,
I'm
look
thank you
forward
all
to
also
very
it.
much.
sorry.
Thank you,
Thank
Dr.
you. Thank you
Siddeem, for
so
giving
much,
us.
and
Yeah.
have a good evening ahead. Thank you.
Yeah. Bye.
Bye. Namaste.